shouting you out there, Snail Hospital. Welcome. I'm going to share a little overview of what you can expect in this snailful hour. We are going to introduce our team to you all. Jann's going to give a presentation about the local slug snails and different projects that we're working on. I'll play a snail game with you and also give you some of my tips on finding and photographing and even maybe drawing snails. And then we'll have an opportunity, plenty of time to ask questions through the chat of Jann. All right, it looks like we're almost at 50 participants here. That's fantastic. So let us introduce ourselves. I'm Maze Connolly. I'm a community science coordinator at the Natural History Museum of Los Angeles County. And I have become increasingly interested in snails and slugs through the course of this project and our slime project at the museum. So I'm excited to share my enthusiasm with you. And I do find snails and especially to be kind of relatable during the pandemic. And I don't know if that's why we have so many snail fans, but I think that whole, you know, retreating into your shell thing, it's something that's kind of speaking to me. Anyway, let everyone else introduce themselves. Let's just go around the slide here, team. Miguel.
Yeah, hi, I'm Miguel Orellana. I'm a co-senior manager of the community science office with Leela, and been at the museum for almost 10 years, which is amazing to think about. And what I love about snails is, I mean, just how hard it is to find them sometimes, and kind of the seasonality of their activity. And as someone who studies mammals and always looking for tracks in the mud, I mean, I'm always kind of running into them when it's wet. And so it's really exciting to find them because I know it's not easy. So yeah, go snails.
That's right, Miguel. Hi, everybody. My name is Jann Vendetti. I am the Associate Curator of Malacology, or the study of mollusks, at the Natural History Museum, LA County. And my research interest when I first got to the museum was marine snails, but I shifted gears a bit, and I ended up becoming very interested in terrestrial snails. And it turns out that there's quite a bit that we don't know about local gastropods. And I'll talk a bit about that and how SnailBlitz and the Slime Project can contribute to what we know and about snails locally and globally, and how you all and your contributions are really instrumental to that. And thanks for being here.
And Leela, our colleague Sam is going to be joining us a little bit late, so we can skip right to you.
Hi, everyone. My name is Leela Higgins. I am co-senior manager of community science with Miguel. I've been at the museum since the end of 2008, and I studied insects for undergrad, so I love some of the more unloved species out there. And I think snails and slugs and insects are in that group of unloved, I mean, very much loved by me. And I think that my favorite thing about snails and slugs is the fact that they're hermaphrodites. They are both male and female at the same time, and they're like non-binary, yay. And they have amazing sexual and mating strategies. And the video of two leopard slugs mating in Life in the Undergrowth with David Attenborough is one of my favorite videos that you should all look up on YouTube. If I can find the link, I'll put it in the chat.
Awesome, thank you very much. I'm going to stop sharing here, and we're going to go on to Jann, who's going to tell us some things about our local snails and slugs.
I'm happy to do that. All right, I hope you can all see that. So I'm going to chat with you just for a bit about some of the research that we do at the museum and some of the projects that we have, and also how humans and human development, meaning like land use development, influences snails and slugs that we see around us. And I'm going to talk about it in 3 different ways. And that is as humans and development and urbanization as a filter and a facilitator and a homogenizer. So some nice words, big words for us tonight. To start with, and this is partly for my friend and colleague Phil, who's in our meeting at this event right now, who is a slug enthusiast, though he doesn't know it yet. This is a slug which its ID I have not yet determined. This is a native California slug that's in the genus Hesperian. Almost nothing is known about this slug. And one of the really fascinating things about this group of organisms is that they are indicators of, if they're native species, they're often indicators of an undisturbed habitat. So if you have a slug or a snail that's native, it's not flying into a new habitat, obviously. It had to be there, and it probably had to be there for quite a long time, evolutionarily speaking. And it had to be in a habitat that was somewhat or almost completely undisturbed. So that often means that there's thick leaf litter, right? So that nobody was leaf blowing or raking that leaf litter away, that it was an undisturbed habitat. And with slugs and snails that are native, you often find a lot of other, sometimes people call them like creepy crawlies, that are really fascinating, generally understudied, and probably very species rich. So things like slender salamanders and insects, roly-polies, harvestmen, spiders, all sorts of things like that. So knowing where these slugs and snails are can teach us about that species in particular, which is my interest, but it also can be an indicator of a particular habitat that's really important to preserve. And you're welcome to jump in and ask me questions as I go if you have any. So just kind of put yourself in the taxonomic frame of mind for this particular group. Mollusks are a phylum. So if you know anything about the taxonomy of invertebrate groups, there are 30-something phyla, 1 phylum, 2 phyla, and mollusks are one of them. Within mollusks, they're the group that we're talking about, snails and slugs, are the gastropods. In all of mollusks, there are about 100,000 species. And within gastropods, or the snails and slugs, marine, freshwater, and terrestrial, there's over 40,000 species, which is huge, a huge group. So here we're talking about just some examples. This is a freshwater snail and some marine snails. And just to kind of put these numbers in perspective for you, there's about between 15 and say 19 or 20,000 species of birds, and about 5,000 species of mammals. So up below the line here at the top of the slide are birds, obviously below are mammals. Give you just another reminder, there's about 40,000 species of terrestrial or land living snails and slugs. So it's at least twice the number of bird species, and more than and 3 or 4 or 5 times the amount of mammals. So it's a really big group. If you were to come to Malacology, you would see our collections. Our collections look like a very well organized closet. If your closet were organized with wooden drawers and in metal cabinets, it should look like this. It should look well organized and very tidy, and this is so that people like myself and other researchers locally and internationally can come here and work, and like a library, that we would be able to find things when we need to. And we think about our collection as a library of biodiversity. The museum has 35 million objects or specimens. It's a lot of different things, and this is a drawer of marine snails, but we have many drawers, as you might imagine, of land snails and slugs. The slime project has been happening since about 2015. We have just over 18,000 observations. So the slime project is snails and slugs living in metropolitan environments. And the project pools together observations that are submitted by people in Southern California. And it started as the only project of its kind really on iNaturalist. And then the SnailBlitz program has been here since the inception of slime, and it has a smaller time frame. And in this project, we really try to get as many observations of snails and slugs as we can, and then these go into the slime project. as well. This project, Slime and SnailBlitz, have been popular in Southern California, but they also have inspired associated projects elsewhere in the US. So there's one called Atlanta Slime. So a colleague of mine in Atlanta has one, and the snails and slugs are far more diverse, or there are many more species in Georgia and the US Southeast. So I don't envy the issue she has in making all those identifications, but that project just started very recently. And then a colleague of mine in the Pacific Northwest at the Burke Museum began a Slime Pacific Northwest project just about a month or so ago. And now they have 37,000 observations, but I think it's because they're pooling all the observations of snails and slugs that were made in iNaturalist instead of them being submitted. But nonetheless, it's a fantastic project. Also, they have far more snails and slugs up there than we do here. And you might imagine why. They get lots of rain. And as Miguel mentioned in the intro, sometimes it's really hard to find snails and slugs here if you don't know where to look, because they're hiding because of our arid temperatures much of the year. Okay, so why should I care? Why should we care about snails and slugs? You all are on this call, so you have your interest in snails and slugs for whatever reason. But as I mentioned from the outset, I'm going to talk about snails and slugs and why you should care, because humans and the way that we change the land that we use and live on can influence snails and slugs and terrestrial gastropods as a filter, facilitator, and homogenizer. So this picture is from San Pedro. These are an Italian or Mediterranean, circum-Mediterranean snail called Theba paisana. These are found in abundance throughout the Palos Verdes Peninsula and elsewhere throughout Los Angeles County and into San Diego and probably northern Mexico. They're introduced. The humans have been a facilitator in introducing these species. So it's not entirely clear exactly how they were introduced, but we do know that they were introduced multiple times. So there are multiple lineages, genetic lineages of these snails living in Southern California and so in Los Angeles County and San Diego County. And it could be that they were brought in for food. These are an escargot or delicacy snail for people around the Mediterranean, in particular in parts of Italy. It's hard to exactly pinpoint how they got here, but there is a historical large Italian-American population in San Pedro, which has an interesting history or possible history with this snail. As a filter, this is a well-known snail to those of us who study the local fauna. For those of you who don't know, this is Helminthoglypta trascae. This is one of your local endemic snails that its range is probably much of Los Angeles County. I think it goes just about a bit outside the county, but largely it's within our county, and it is found here and nowhere else. And it doesn't live where there is human development, where there has been a loss of leaf litter and a loss of habitat. It has to have leaf litter, oak woodland, and other habitats. It's very happy living there, and it can estivate or go sort of to snail dormancy or snail sleep for quite a long time until conditions get better. You can often find it by looking under leaf litter. The place that I usually find it is in or under rotting logs. That's where a lot of really amazing biodiversity is. So if you find a habitat where there are rotting logs and you gently turn them over, sort of like if you're in the intertidal, you turn over a rock, you look at what's underneath it, and then you put it back so that those little creatures can still stay alive. So these are our friend, the Trask shoulderband snail. And the reason that it's a filter, so some years ago, this is in 2017, I was at Glendale Community College at teaching a class, and I was walking back to my car at 10:24 p.m. and I saw this little snail friend crawling right next to the parking structure. And I was surprised. And Glendale, this community college, is up in the San Rafael Hills. And they live there. So there's a population that I was unaware of, of Helminthoglypta trascae living up there. And then a couple of years later, that particular patch of dirt where I photographed this specimen was paved over. So that little habitat where the snail existed was gone. So this is an example of a filter, meaning that you can have an environment, but if you change that environment, humans have filtered out the species that can live there, such that now the species that are living there are introduced. This is one actually just from a few days ago at a different part of campus. there's still some Helminthic living there. They're just not in that particular spot where I found them earlier. This is what it looks like if you were to look in our collection, Helminthic. We have hundreds of records based in specimens in our collection, and we can use those to figure out where these snails used to be and compare it with what habitats look like now. So as just an example, this is a specimen from 1936 that was collected on 24th Street, Hermosa Beach. In the 1940s, this was 1936. In the 1950s, all of these houses were built in Hermosa Beach on 24th Street. So the habitat is completely gone for this snail, right? This is a filter, an example of a filter. Now the species that are there are the garden snail, 3-band slug called Amigo limax, Drosera reticulatum, the milky slug, and a species of Oxychilus. And you can see, if you all know iNaturalist, or if you don't, this little IN, pink IN in the corner of the image means introduced. So the species that are there now are all introduced, which is another way of thinking of this filter and facilitator, having all of these introduced plantings that came from Home Depot or Osh or wherever, or different nurseries around Southern California and elsewhere, brought in with them all these snails. So nobody's bringing in these slugs to eat them. Likely this garden snail isn't coming in as a food snail. It's coming in accidentally. Also as a homogenizer, just to give you a sense of this is a really fantastic picture from iNaturalist. It's from southern Spain. This is from southern Spain, though we have all of the species in Southern California. We have all of these species. So in yellow is Cornea spursum, that's our common European garden snail. In pink is Otellolactea, and then in blue is Theba paisana. And Otellolactea, just to give you a quick little story, some students and I are working on this species. Otellolactea is found in at least a dozen separate locations with disparate non-touching populations throughout Los Angeles and into San Diego County. And doing some research, genetic research on them, we found that much like Theba paisana, that Italian snail that I was mentioning in San Pedro with the Italian Americans, that it looks like this snail has a very similar history, that people possibly brought it in as a food snail, but it moved around and it got distributed throughout the US and elsewhere. And there are multiple lineages. So it has come into Southern California and elsewhere multiple times. And this is just an iNaturalist screenshot of some of the locations. These teardrop shapes are locations where the species was found. And then you can see that it's also found in parts of Texas. And then this is our friend, just as a comparison, these are Theba paisana, those other snails that were brought in and have a similar history to these Otellolactea. And these are Theba paisana snails being sold for food in Sicily. Just so you can see, there's a lot of interest, a lot of food interest in these snails. So it's quite conceivable that they were brought in as a food item. And just a few short stories about some of the introduced snails that you are likely to find in Southern California. Cornea spursum, as I mentioned, is a common European garden snail. It's been in Southern California for more than 100 years. One story goes that it was brought in by a Frenchman who actually asked via mail someone in his family to send him snails, and they did, and this gentleman wanted to raise them as escargot, and he made some sort of mesh and didn't realize that the little babies would be small enough to crawl out of the mesh as babies. So then they became, they escaped their enclosure, and that was, then they became populated all throughout California and really problematic in citrus groves, when citrus groves were much of what Southern California habitats were. Xerotricha conspircata, this was a paper that some colleagues and I, including a number of community scientists, published together in 2018. This is a European species that was found in multiple locations throughout Southern California. Pretty small, about the size of your pinky nail. It was the first record of this species in certain counties in California. Pupoides albilabris, this is a southern US, southeastern US species that was found as an introduced species for a first record in Southern California by a community scientist. Cocosella barbara, the same thing, this Mediterranean species. If you'll see a trend, many species that we have here are common to the circum-Mediterranean region. And when they end up here, usually accidentally or sometimes on purpose, they end up doing very well because their climate is very similar in certain parts of Los Angeles. So they do very well. They can estivate or go into snail sleep for quite a long time, which means that they can withstand our very hot summers and very little rain. This is some examples of Coccolus alabarbara throughout Southern California. It's a tiny little snail. You can see a finger for scale. Their distribution goes all the way up into Santa Barbara. This is Amigo limax. There are 2 species of this. So a tiny, very, very short story. When I was starting to study slugs, terrestrial slugs, I wanted an example of some slugs to look at, and where the campus that I was working on, I thought, oh gosh, I don't have any samples with me right now. Let me go look under an electrical box or a sprinkler box, if you know what those are. In most developed areas to control irrigation. I lifted one of those up, pulled some slugs out, and I remember thinking to myself, you know, I cannot tell the difference. People here say that there are possibly two species, Nyctelius and Valentianus of this genus Amigo limax, and I should be able to tell the difference, and I can't. And I thought, oh gosh, what's wrong with me? I can't tell the difference between these. And then I did some genetic work on them and determined that we have both here, and they're almost pretty much indistinguishable by phenotype. So it means what they look like, they can look like each other, they can look different from each other, they have almost no distinguishing characteristics that you can use to differentiate them. So when we find an Amigo limax looking slug, we just call it, on iNaturalist, I just call it Amigo limax, because there's no way to tell which species it is of these two, Valentianus and Nyctelius. And then these are some snail and slug friends that we hope to not have in Southern California. So these are problematic invasives in different parts of the world. And invasive just means that they end up here for whatever reason, however they got here, and it becomes problematic for species around them. So they can be vectors of disease, they can eat native plants, they can outcompete native species. For whatever reason, that introduced species is considered problematic, thus invasive. So if you see any of these snails or slugs, please post them post haste, post them right away to iNaturalist so we can get a handle on where they are. But the observations that community scientists make in all of these projects are very necessary, because much of my research, and anyone else who's doing research on native or introduced taxa of terrestrial gastropods, is quite reliant on knowing where those individuals are and how their populations are related, if they're interbreeding with each other or not. And with that, I'll wrap up Maze, and I'm happy to answer questions now or later.
Thank you so much, Jann. I'm sure that inspired a lot of questions, and we'll be sure to leave plenty of time for questions. I know I probably have some younger folks in our meetup today, and those of you also who like to join in. Everyone, please, I'm going to do a very quick round of 2 truths and a lie. So here's how we play the game. I'll read you 3 statements. You tell me which number by typing in the chat is the lie. I'll tell you if you're right. 2 truths and a lie. Inside those smooshy bodies, snails and slugs have a heart, kidney, lung, and intestine. If you think that's a lie, type number 1 into the chat. The average garden snail has approximately 400 teeth. If you think that's a lie, type it into the chat, number 2. After mating, slugs go their separate ways and then both lay fertilized eggs. If you think that's my lie, type in number 3. I'm seeing a lot. Oh, I don't know. Is there a front runner? Can you tell, Jann, in the chat?
It's moving a little too fast.
Miguel or Leela, it seems like a lot of people think number 1 is the lie. That's what
I was seeing a lot of 2s.
Number 2.
And then I think a lot of 1s started happening too.
It's hard to believe that there could be that much of it, either teeth or those organs inside of a snail or a slug. Are you ready for the answer? Number 2 was the lie, but not probably for the reason that you thought. The average garden snail has 14,000 teeth. 14,000. How is that possible, Jan?
Yeah,
Here.
I usually tell people that a snail tongue, sort of the best way to think about it, is it's sort of like a cat tongue. If you've ever been licked by a cat, they have a really rough tongue, and snails and slugs have that. So they have one jaw, typically, not always. They typically have one jaw, and then they have this structure called a radula that's filled with teeth, and those teeth can be different sizes and shapes. Some sea slugs have very few. Some have evolved to have none, but most land slugs and snails have many teeth along what that radula is sometimes called a ribbon. So a ribbon of scraping teeth, and what they do when they're eating is they rasp or scrape whatever it is that they're eating, and then little bits of food get caught or stuck on that those many little teeth, and then they swallow that. So it's even something you can see if you have a snail or a slug and you have the opportunity to watch it on a piece of glass or a window, or even just very carefully watch it eating something. Sometimes you can see the radula come out, and you can't see the teeth very clearly, but it may look like a rough structure, and what you're seeing are all those little
that could explain why you can sometimes hear a snail eating, right?
Yes, indeed. Yeah, that's absolutely right. Yeah.
Sound.
They're doing a lot of work.
That's me impersonating an eating snail. You're welcome.
A lot of work.
Okay, are you ready for another question? Another 2 truths and a lie. Number 1, mating snails sometimes shoot each other in the head with darts. Number 2, you can only find banana slugs north of San Luis Obispo County. Number 3, snails grow their own shells. Which one is the lie? I need some music to go with the contest. Oh, I'm seeing a lot of number 2s. No, what, number 4, wait a minute. Number 1, number 2, number 3, I'm getting them all. Okay, what do you think, Jan? Number 2 was the lie. 2 banana slug observations have already been added to SnailBlitz 2022. Where might somebody find a banana slug in Southern California?
I have never found one, but there apparently is a species, perhaps still undescribed, on Palomar Mountain in San Diego County. Must be San Diego County. And there are, if you are in certain habitats, maybe just, yeah, I think that's right. Yeah, that I'm trying to think of somewhere else. Maybe
That's
very,
amazing.
yeah. Probably
I want to find a banana slug. What should our guests do if they find a wild banana slug in San Diego County?
Yeah, well, you would likely be in a remarkably old and undisturbed habitat, I would think. And the group of banana slugs that are sort of resident on the mountain are kind of holdovers from when when banana slugs had a larger population, and then now they're restricted to just that particular location, and they have to be in an environment that's more wet than what we would typically think of as much of the habitat that we interact with most of the time. So
yeah, this banana slug in this photo is actually one that I took, but it was not in Southern California. So up in the redwoods where it was very wet, where you would expect to see them, where they can have
crawling around, right? You don't have to look as hard. You have to look under
they
rotting
even have
things.
a mascot right up there.
That's right, Santa Cruz. That's right.
As the banana slug mascot. Okay, I'm going to give you the last 2 truths and a lie. Number 1, most snails mate for life. If you think that is the lie, please press 1. Oh no! Oh, phew! That went the wrong way. How did that happen? Okay, sorry about that, everyone. I thought I just blew my answers there. Number 2, snails poop and breathe out of the same hole. Is that a truth or a lie? If it's a lie, put number 2. Number 3, in an international search for a mate, an international search for a mate was conducted on behalf of a left-handed snail named Jeremy. If you think that's the lie, press number 3. All right, let's see.
I just want to let you know, Maze, we put it in a poll.
I see, that was clever. Thank you very much. I wish I had thought of that in advance so I could really see. So I think so far.
Maze, could this be 2 lies and a truth?
Uh-oh.
Is it, Jann? Did I get something wrong?
Yes, I think that this is 2 lies and one is true.
Oh,
I can tell you once we're done.
I should have had a fact checker.
So far, we've got 62% saying that the lie is most snails mate for life. 24% snails poop and breathe out of the same hole. 17% there was an international search for a mate for the left-handed snail, sinistral, right?
That's correct.
Snail Jeremy. Oh, Jeremy.
Oh, I think you might have just given that away there, Leela. But I think we're at the end of our voting here. So I'm going right to the answers, which are wrong anyway, according to according to Jann.
Well, I can walk you through that, everybody. Maybe it was Maze's trick question.
Yes.
It was Maze's trick question. So, Maze, do you want to talk about Jeremy?
Well, actually, first, I want to say I'm pretty sure that if I got one wrong, it was number 2. So
That's
number
correct.
1 was definitely a lie.
Yes.
So there are 2 lies.
Yes, there were 2 lies. So snails and slugs don't mate for life, and they're very opportunistic, and they breathe out of a pneumostome, which is an air hole. And
so that's what's indicated right there in that little illustration.
Yes, okay. And their body is twisted in a snail, and also in a slug, but it's more obvious in a snail, I guess. Their body is twisted inside so that if this is a snail, right, and the head is here and the backside is here, it's not pooping out of this. Its body is all contorted and twisted. It's called torsion. Its body has been twisted, and it's twisted within the shell such that its reproductive and its digestive system are through the shell and up into that part of the body, and in a slug too, it twists as well, and then it comes out the side. So a snail will poop right next to its head.
Okay. I think I have seen snail poop around the pneumostome. So that's
probably, yeah, it's close, right? It's very close because they're breathing, let's say, I'm not necessarily in the best orientation here, but if they're breathing through this hole here, then they're pooping out of a hole that's very close.
Okay. All right. My bad. But I also hope that you can appreciate this picture of Jeremy, who was a famous sinistral snail. Now, that means that Jeremy's shell coils basically like if you were looking at a boat and you would say the port side and the starboard side, right? All the snails that you will normally find have their outside of their coil on their port side.
On their right, yeah.
Their right-hand side.
If you have it opening, the shell opening towards you, the aperture or the opening is on the right. In Jeremy and other sinistral or lefty snails, the opening, it curls the other way such that the aperture, the opening where the snail comes out is on the left. And the best way is if you're confused, if you look at that image and you imagine turning that top snail towards you, like with the opening towards you, that opening would be on the left.
Right.
Of the animal or of the shell.
And the one underneath it is like every snail you will probably ever see in your life.
Yes.
Right. Yes. Or at least this particular species, or the species that we have here. And we did have somebody find a rare sinistral snail in Los Angeles, right? It was at the museum for a while.
Yes, it
and
was.
It was named Sandy.
That's right. That's right.
Jan,
Yes.
so how many introduced snail species do you think there are, either in California or LA County?
Ooh. LA County, I think that there are at least a dozen.
Hmm.
I looked up those numbers somewhat recently, and it escapes me right now the exact number, more than a dozen. In all of California, it's 20 something at least, 20 something.
Right.
There are some species too that are in species complexes such that they're difficult to tell, just like that I was talking about that slug, that long name, Amigo limax, and there's two species and they look very similar, and so it's hard to tell them apart. So sometimes if on iNaturalist, it's just identified as Amigo limax, but we do have two species, and that's the case for probably a number of species within a number of different genera of introduced species. And then native species is tough. We have likely we're quite undercounting or underestimating the number that we have in California because many of them aren't being studied, like that little slug that I showed at the beginning of my slides. It's possible that that's an undescribed species because very few people have worked on it. And what used to be described based on shells, slugs don't have those shells. So often they were described based on their morphology or what they looked like. And it's very possible that there are multiple species that look the same or very similar, but genetically they're dissimilar and they're distantly related enough that we would consider them separate species. So ballpark, I don't know, 75 native species.
Wow.
I mean, I should be a little bit sharper about that, but it's probably an undercount. Yeah.
Wow, that's so
Thank
many.
you for that question.
Listen, I'd like, I have a feeling, I have a few more slides and I have a feeling we're going to have a lot of questions. So I'm going to kind of race through these a little bit. And I'd like to ask, if you have questions, would you please either type them into the chat or put an asterisk in the chat, and we'll try to call on you in order. And I'm just going to share a couple of things here, like ways to find them, especially during this dry year when you might go to a place where you've seen lots of snails in the past and you're not finding as many as usual. Jann is advising us to all look under leaf litter, especially, or under rocks. And if you can't find them in the urban environments, look around irrigation, places that are irrigated.
That's absolutely right.
Right.
Yep.
And then one other point that's not on this slide is on north facing slopes. There'll be the cooler and wetter side, the cooler and wetter places. And then when you're photographing one for a snail, if it's not alive, try to get a picture of both sides of the shell and also that aperture, that opening, if you're just finding a snail shell, or if there's a living snail and it is stretched out like that, try to get a picture of its extended neck. I know it's not really technically a neck, right? And then I think snail portraiture is also a wonderful thing. So I would recommend, if you want to get an artful picture of your snail, that you consider putting it in a not too contrasty lighting by gently moving the leaf into a sort of a somewhat shaded area, and also just wait, because it will definitely, if it's awake enough, give you lots of good poses. And then Jann, I don't have pictures here, but basically for our slug observations, for you to be able to ID a slug, we want to see the top view that shows the mantle of the slug, right? Which is basically the back of the head.
Yeah.
If you can gently, maybe using a leaf or a stick, flip it over to get the foot.
In some species, that is a diagnostic character, or could be. in not all species, but yeah, if you have the opportunity to. But the pneumostome and the mantle is a good one. So if you get it from the breathing pore,
Okay.
breathing hole, that's very helpful. So that and the pattern on the body of the slug is often enough. Within the smallish number that we have in Southern California, it's usually enough to get it to species, and if we can't do that, to genus.
Great. So those breathing holes have different positions on different species,
Yes.
like they're located in different places.
Yep. And they sometimes have a
That helps.
ring around them that looks white or it doesn't, and sometimes that's diagnostic as well.
Oh, I was going to show you how to draw a snail, how I like to draw a snail. I just wanted to point out a couple of common pitfalls in case there are some artists here who are enjoying all the snail cartoons. The thing that I noticed people doing the most when they're drawing a snail just as a cartoon is putting the shell on backwards. So if you really look at a snail, and like we were talking about on Sandy, the opening of the snail should be toward the head. Might seem obvious if you've looked at a lot of snails in person, but you'll see now that I've mentioned it that a lot of people draw them on backward. The other thing that's a really common thing to see is they've got these cool, crazy eyes at the end of these eye stalks, but a lot of people just think of those as horns. And so they'll draw a little smiley face down where those other two tentacles are. And that's the last thing too, the tentacles. People often forget those tentacles down there at the very bottom. So those are my hot tips for drawing a snail or slug. And now let's see, I'm sure we've got plenty of questions. I'm going to stop my share. And let's see what we got in the chat. And Sam, who we mentioned earlier, my colleague who couldn't join us at the beginning, is here, and they've got some chat questions saved up for when Jan is ready. Sam, are you in a position where you can chime in and share some of those questions?
I sure can. Can you hear me?
Yes. Hi, Sam.
Hi, y'all. This is so cool. All right, let's jump into it because folks had some really great questions. And yes, I love Maze's drawings too. Anyway, Caitlin asks, why do some marine gastropods have fewer / none teeth than terrestrial? Is it due to different diets?
Yes. So the most famous example, I could talk about this for a while, but we've got limited time. The most famous example of reduction in the radula, that structure, that like tongue structure, is in cone snails. So if you know anything about cone snails, they look sort of like a cone. They have a really thin aperture where the animal pulls in, and they're predatory. So they eat other snails, marine worms, they can eat fish, and there are more than 500 species of cone snails. They're a really species-rich, particularly species-rich group, and they've evolved to have a reduction in the radula such that their radula, instead of being a tongue full of many, many teeth, is one single teeth that is like a harpoon that they can shoot out of their body, and with it comes a particular kind of toxin. This is a bigger and more amazing story, but there are chemists and biochemists who have studied these toxins and have come up with therapeutics for human diseases and pain relief based on these conotoxins. And they're called conotoxins because they're from cone snails. And anyway, they use that toxin shot through this little harpoon radula to immobilize their prey and then eat it. So that's the short answer, but I could go on and on. It's a fascinating system to work in. I don't work in it, but if you wanted to look up anything about it, it's cone snails. And if you look up cone snails eating, sometimes they have to slow down the video because it's so fast that you can't see it when it's filmed. It's too fast to see.
That's different from the love darts that
yes,
we were asking about earlier.
Yes,
right?
Yes, it is different. Yes.
And speaking of which, as much as I would love to hear more about that, this is a good one too. Martine asks, how do they throw darts at each other?
They also have a dart sac in their body that as they're mating, they have, remember I mentioned that I'm trying to display this with my hands. Their body is contorted such that they've got all of this interesting anatomy at their disposal right near their head, right to the side of their head. And that includes this, not all snails have this, a dart, like it looks like a little dart, and they can eject it. And they may have, different species may have different darts, such that if you happened upon a particular dart, you may be able to tell the species based just on the dart, the morphology or the shape and size of that dart. Anyway, they shoot that at their partner, and one hypothesis is that there are hormones in that dart that make that partner, mating partner, more likely to lay more eggs, or may lay eggs repeat. I can't remember the exact interpretation, but it facilitates egg laying such that the partner that shoots the dart is a parent of those offspring that are laid by the partner, if that makes sense. It's amazing. It's amazing.
Wild.
I've never seen it.
That is wild. It's really incredible to me that there is just so much, variety, anatomy, behavior, and everything, and something that really probably just a few years ago I thought was just like a little lump of slime.
I can understand that. Yep, they are so very weird and fascinating and so different from us. But then, like you said, Maze, earlier, in some ways, you can sort of find solace in the snail and its slow pace of life, and perhaps some of its challenges. But it is a very different creature, and there's such a tremendous amount of diversity that it is really mind-boggling. And earlier, I lost my math skills while I was confounded by the number of mollusks, and I think I said that there were 2 or 3 more snails than mammals times more snails, but it's more like 7 times more land snail species than mammals. Just to throw that out there. No offense, Miguel, my colleague who studies mammals.
I see that some folks are already leaving us, and so I just want to make sure we mention that our SnailBlitz project is still going until the end of March. So if you get an opportunity to photograph some snails between now and the end of March and upload them to iNaturalist, you will definitely be contributing to SnailBlitz if you're in the Southern California region. Now, this is just land snails and slugs in Southern California.
What else can I answer? Anything else?
Yes, we actually have a bunch of questions. Lorena asks, why do they close their breathing hole? I'm guessing it has to do with conserving moisture, but are they holding their breath while it's closed?
That is a great question, and I've never really thought about that. They don't close their breathing hole all that much, slugs at least, but presumably, yeah, I guess they would have to be holding their breath. I guess they would, but I've never thought about it. And slugs, but snails more sort of obviously can go into this snail sleep or dormancy called estivation, and they still breathe, but they slow their metabolism down quite a bit so that they can operate, they can live maybe in their entire life, which could be, let's say, 5 years for a land snail species or individual. Let's just say that 90% of its life could be estivating, that most of its life is just in this sort of snail torpor. But yeah, your question, I guess they're holding their breath. I guess they are. I wish an awesome thing to think about.
Right? I wish I could spend 90% of my life estivating. I got a lot of questions. Sorry.
I wanted to mention also one more thing before you sign off. I'm going to send you a link to a survey that we would love you to fill out about this presentation. And I also have printed a couple of little vinyl stickers for all of you. If you're interested, please email me your mailing address and I will send them to you. It's not for the mailing list. It's just for me to send you the stickers, and I'll put my email in the chat. And you can see a link in there also for the program survey. You can also see a link for the bingo card, and I'll put my email in there too. My email is also at the very end of the survey. All the questions are optional, so if you are just looking for my email, it is on there as well.
Okay, next question. This is a good one from Sophie. If Jeremy has babies with another sinistral snail, will the baby also be sinistral?
This is an excellent question, and I was at a mollusk conference, and I had the opportunity to ask Jeremy's, if you will, owner, if Jeremy could have such a thing. He's a very prominent malacologist, and he was studying that exact phenomenon, studying that, and the genetics are much more complicated than just lefty snail and lefty snail have babies that are lefty snails. So I would have thought, oh, a simple system would be like that. And that would be like my default assumption, but it is not the case. It is a much more complicated scenario that I cannot explain right now. He explained it to me, and it took me a while to understand exactly what he was talking about. It's a really complicated, well, it's somewhat complicated. I'm sure if it were written out, we would all be able to understand it, but it was not as straightforward as lefty snail has babies with lefty snail and has lefty babies. Or excuse me, I think you follow what I'm saying. Yeah, so, but most of the time, right, what we call dextral snails, they mate with dextral snails, and then they have dextral babies. But the issue with Jeremy is that because a dextral snail's snail shell and it's not a his snail were oriented differently, the reproductive parts would not line up properly. So for Jeremy to have babies with anyone, it would have to be another lefty snail. But like I said, the genetics of what their baby's snail coiling is not particularly straightforward.
Ah, okay. Snails are so complicated. Okay, so Alex says, the upper Newport Back Bay is covered with milk snails in spring and summer. Today we only found one broken shell. What's hiding? Is it eggs? Are they in the soil?
That's a great question. I'm so glad you're here, by the way. Let's see. I asked a colleague also recently to see if they could find some, and she couldn't find any of those milk snails where she is. And I don't know the answer to it. They're likely not burrowing. It could be that that population was taken out by a predator. It's possible. Native California ground squirrels will often eat them. It's possible other birds, I haven't seen birds eat them, but they certainly could. And then maybe raccoons and rats and other mammals that we have here might eat them. So it could just be that particular population was eaten up. I was recently in Long Beach looking for some, and the population that was on there identified on iNaturalist, I only found shells also. And they were pretty numerous in iNaturalist, but I looked hard and I just found shells. And where you find one, you typically find many. So it must have been that something ate them, or it could be that there was an effort to remove them, and they were removed by hand picking, or maybe there was a Sluggo is a molluscicide. So somebody might have set out food for them to eat that would make them die. So that's possible that happened, that you only have the shells behind, my guess.
Okay, and here's a good question from Marike. I'm sorry if I pronounced that wrong, but they say, I normally try not to disturb critters I'm taking pictures of. How robust are snail slugs, i.e. if I am gentle, does it put a lot of stress on them to be flipped over, etc.?
Yep, that's a great question. They are very robust. So they can survive that. Any species that we have here are quite robust and can survive that. And just as sort of an interesting factoid, if any of you are familiar with South Korean and other East Asian snail mucin like skin treatments, there's very expensive snail skin lotions and things that are considered anti-aging and have all these sort of remarkable properties. And to get the snail to make slime that is put into these cosmeceuticals, sometimes they're called. They are put into this, and the patent says this. They are put into a modified salad spinner, and they're centrifuged and agitated, but they live and they're fine. But they just make a lot of slime because they're very upset about being spun around in this, presumably very unfamiliar way. They produce a lot of slime, and then that slime is filtered and put into these lotions and whatnot. But the snails survive. So anyway, that's just to make the point that most snails and slugs, if you manipulate them for a photo, that's not enough to disrupt them and kill them. They're usually good photo subjects.
I feel bad for laughing. That was pretty wild. Centrifuging
They have a story to tell. The ones in the salad spinner. Yes, maybe don't try that. They've got some stories to tell their friends.
Okay, Laura has a great question. Do you think populations of banana slugs could be found in the Angeles National Forest or other areas of Southern California?
Wouldn't that be amazing? I think if they were there, we would know. But then that could be I don't know. That could be me saying that, assuming that we've really trekked about throughout the San Gabriels and we know every inch of that, habitat. That they haven't been found, it does make me think that they're not there. But could they live there? I mean, that's a great question. I don't know the difference between, I don't know why they would be on Palomar Mountain and not our San Gabriel Mountains. I don't know. Someone knows. Someone likely knows. That person is not me, but it doesn't sound completely inconceivable. But I think if they were there, we would know.
Okay, awesome. Maybe one of us will find one up there. Joseph asks, is it true that evolutionarily slugs are a kind of snail?
That's right. So yes. So thank you for this question. So slugs are not a snail that's like lost its shell in life, like during its life. It is a lineage of snail that has lost its shell evolutionarily. So over many millions of years, that particular lineage, it was beneficial for there to be a reduction in the shell or a mutation in which there wasn't a shell, and that lineage survived regardless. And this has happened multiple times within all of gastropods. So a slug form has evolved multiple times. Most of the snails and slugs that we have in Southern California are all within the same evolutionarily related group. It's like a monophyletic group, if that matters to you. It shares a common ancestor, but there are multiple different lineages that are slug form. And that means that 500 plus million years ago, the presumed ancestor of gastropods, as far as we know, was shelled.
Wow.
Though, just to say, it is easier to find things that have shells than things that don't, right? So it is, so shells fossilize and soft bits don't typically, but there is a small shelly fauna that's it's older than 500 million years. And some of those fossils are interpreted to be ancestral gastropods, or certainly ancestral mollusks. And the ancestral mollusk has a shell, the way that it's interpreted by malacologists.
Wow.
Thank you so much, Jann, for answering all of these questions. I see we're already past 7 o'clock. I'm sorry, I disappeared for a minute there to try to fix our permissions. I would love it if any and all of you are willing to fill out our survey and share your thoughts on this program. The link is in the chat. And I will be sending out, we've recorded this program, I'll send it out as a link, if everything goes as planned, to you and those of the people who signed up and didn't get to be a part of the program. Jann, do you have any parting snail bits to share?
Oh, just that I'm really so happy to see so many of you and some people who are working on projects with me and have interacted with me on iNaturalist. It really is an honor to be able to see you and interact with you. And thank you for your interest and your participation in SnailBlitz and Slime Project, or just in general, this appreciation of biodiversity. I think it's a remarkable community to be in, and I'm really just so grateful.